Today on Flipping Science, we're going to be talking about proteins. So what do you need to be able to do? So you need to be able to identify the amide group and figure out the structural formula of any monomers that make up a section of a protein. You need to be able to figure out where hydrogen bonding can occur between protein chains and between protein chains and water. You need to explain why enzymes don't work when the spatial arrangement is altered, so when they change shape, and also explain the effect of pH and temperature on protein chains. So what are proteins? Proteins are made up of amino acids. And you join the amino acids together with peptide bonds or amide bonds. They're known as polyamides. So if we look at a section of protein down here, we can see we have our amino acid and they're joining together, and when they join together they make up these amide groups. So you get C double bond O NH, and that tells you that you've got an amide group there. So if you need to figure out the structure of monomers, if you're given a section of protein, first thing you do is find the peptide bonds. Then you need to cut them, because that tells you where monomer 1, 2 and 3 are, in this case for this example. You cut at the amide bond between the nitrogen and the carbon. When you do that, you separate it out. You then need to just finish off the amino acids by making sure that any carboxyl groups has a hydrogen and any amine groups has its nitrogens. So it should be C double bond OOH for the carboxyl group and NH2 for the amino group. So you need to do that to finish off the structure of what the amino acid would have been. So hydrogen bonding is very important. It can bind two sections of a single protein, so two strands of a single protein together if they loop around. You can also get hydrogen bonding between the protein chains and water molecules. So if you're looking at a protein chain, you need to look for sites where hydrogen bonding can occur. So remember, hydrogen bonding, it's between when you have nitrogen hydrogen, oxygen hydrogen and fluorine hydrogen bonds, that's where you can get hydrogen bonding occurring. So if we look at our example chain down here, we've got nitrogen hydrogen bond here and we have an oxygen on the other side. That's from a amide group over here, but we can still get hydrogen bonding between them. So that's what you're looking for when you're looking for figuring out where the hydrogen bonding will occur between chains or between chains and water. The shape of proteins is very important because the shape of the protein determines what the protein does. Some proteins act as enzymes, and what that means is they can speed up chemical reactions. So they're biological catalysts, basically, that's what we call them. The way an enzyme works is it's down to its shape. In biology, we call the reactants in a chemical reaction a substrate. So the enzyme can change the substrate. So here's an example showing that. So here we have our enzyme over here and we have our substrate, so that's our reactant. The enzyme and the substrate bond together, just with weak bonds, it's only temporary bonds. The enzyme changes shape slightly, and that forces the substrate. It can force bonds to break, it can also force bonds to be made. When the enzyme changes shape, so it forces those bonds to either break or be made. In this case, they're being broken. We're breaking up our substrate into two products. And then the products are released from the enzyme, so they undock from the enzyme, and the enzyme is ready to go again. So a single enzyme can do many, many reactions per second. They're very, very efficient ways of increasing the rate of chemical reactions. So how do they work? So here we have our protein, and here we can see there's this empty space in the side of the protein, and that's where the substrate can bind. So when the substrate comes in, and we can see over here the enzyme substrate complex, we can see the substrate on the inside, and we can see it's fitting into the active site. Once that docks, then like I said, the enzyme changes shape slightly, and that forces bonds to either break or be made, and then you get your products being produced. So because proteins, the shape of those enzymes is very, very important, so the shape of the active site that we had back here is very, very important. If you change the shape of the enzyme, you change its effectiveness. You can stop an enzyme from working. And two ways of changing the shape are by changing pH and by changing temperature. So pH can change the bonding between positively charged amino groups and negatively charged carboxyl groups. When you put these in solution, they can ionize, and then you can get attraction between the positive group over here and the negative group over here. If you reduce the pH, so you make it more acidic, what happens is the extra hydrogen ions that are in the solution, they can bond with the negative part over here. And you get back to having your full carboxylic acid group over here, your full carboxylate group. If you increase the pH, you are increasing the concentration of hydroxide ions, and those hydroxide ions can take hydrogen ions away from this positively charged amino group over here. And that returns it back to an NH2 group, so it doesn't have a charge anymore. So because they don't have charges, what that means is the attraction between them is gone, basically. So you're not going to get any attraction between the negatively charged group over here and the positively charged group over here. So what that means is you stop ionic bonding from occurring between those parts of the chain, and that can cause the chain to unravel. And that will change the shape of the molecule. So the effective temperature, what happens is when you heat up an enzyme above 50 degrees, you're going to be breaking most of the secondary bonds that are occurring. So you're going to be breaking hydrogen bonds, you're going to be breaking dipole-dipole bonds, and you're also going to be reducing the effectiveness of dispersion forces. So what that means again is the shape of the enzyme, or the shape of the protein, will start to unravel. So rather than having nice tight swirls and bonding between parts of the protein, it starts to unravel into a big strand. And basically you don't have your active site anymore, so the enzyme doesn't work. So let's add some example questions. So here we have a question that says, when a potato is cut and exposed to oxygen, the potato starts to brown, and that's catalyzed by an enzyme known as PPO. PPO is formed from amino acids such as serine and glycine, which we've shown the structure of. Then it says, draw the structural formula of a section of PPO that is formed from one molecule of serine and one molecule of glycine. So we need to basically join these together. I'm going to start with, so I like to have my amino group on the left-hand side, so I'm going to start there. So I'm going to start with my nitrogen here. I'm going to have that blank because that could be going off to the next amino acid. That's joined to a carbon, which is joined to a hydrogen. Then we have our variable group here. So we're going to put that down here. So we've got CH2OH. So that's our variable part of the amino acid. And that's joined to a carbon that forms the double bond O. So that's part of the carboxyl group. So now we have C double bond O to N to H. And now we're going to draw the glycine molecule. So we have going to a carbon, going to a hydrogen. Now in glycine there's no side chain, it's just a hydrogen atom. And then we need to draw our carboxylic acid group to O, and then that will bond to the next amino acid. The next part of the question says, the browning reaction can be prevented in a number of ways. Explain how placing cut potato in boiling water for a short time prevents the browning reaction. So what I would say here is the increase in temperature is going to change the shape of the PPO protein molecule. The active site of the enzyme, the shape of the active site of the enzyme will change. That means the PPO won't be effective at browning anymore. So the effectiveness of the protein is reduced by increasing the temperature of the protein and breaking those secondary interactions so that its shape changes. The key is the shape will change when you increase the temperature, and that will reduce the effectiveness of the protein. Next question here. So this is a nice big long question, and it kind of links in with what we just had before. So this was an extended response question, so it says you should take about 10 minutes to do it. So we're given a structure of one enzyme, so a section of a structure of one enzyme I should say. Then it says explain how high temperatures and high pH would affect the ability of this enzyme to function. So we'll start with temperature again. So in terms of temperature, when we increase the temperature, we reduce the effectiveness of secondary interactions between parts of the protein. That means that hydrogen bonding between parts of the protein, dispersion forces acting between parts of the protein, dipole-dipole interactions occurring between parts of the protein, and even ionic bonding between parts of the protein will start to reduce in effectiveness. And that will lead to a change in the shape of the protein, and therefore the effectiveness of the enzyme will reduce. So its ability to function will be reduced by the change in shape caused by the high temperatures, breaking the secondary interactions between parts of the protein. So the other part is talking about pH, and if we have a look at the section that they gave us, helpfully they gave us this positively charged amino group over here. So what that means is if we were to increase the pH of this solution that the enzyme would be found in, hydroxide ions that are present caused by the increase in pH, so it's becoming more basic, they are going to remove the extra hydrogen that's attached to that nitrogen. And when that happens, that will reduce the charge there, so there won't be a charge anymore. It'll just be an NH2 group rather than an NH3 plus group. What that means is that any ionic bonding that occurs between parts of the chain that involve this positively charged amino group over here, they won't occur anymore. So again, the shape of the enzyme is going to change and its effectiveness is going to be reduced. So by making the solution more basic, by increasing the pH, the hydrogen ion that's attached here will be removed. It will be part of the, it will be basically neutralized essentially. It will become part of a water molecule that's formed from the hydroxide ion and that hydrogen ion. The positive charge here won't be present anymore, so any attraction between this positively charged part of the enzyme and a negatively charged part of the enzyme won't occur anymore, and therefore the shape will change. The shape changes, that reduces the effectiveness of the protein. So here's another question. It says, structures of two protein chains in here are shown in the diagram below. Draw one hydrogen bond that forms between the two protein chains. I think the easiest one is going to be between this hydrogen here and this oxygen here. So I'm going to put in my dotted line. That's where we're going to get one of the hydrogen bonds that's occurring. It also says indicate the partial charges. So the oxygen is going to be delta negative and the hydrogen is going to be delta positive. So that's it for Flipping Science today. See ya.